Okay, some more key terms that you really need to have a handle on to be successful with these Hardy-Weinberg math problems. Remember that a population is a group of individuals of the same species that live in the same space. So a gene pool represents all of the alleles collectively in a population. So say you have a herd of javelina, that's a species we know very well here in Phoenix, and each javelina has a genotype composed of two alleles for this particular gene. If you take all of those alleles, all of those bees, and you dump them into a little Mr. Turtle pool, that is your gene pool. So hugely important in Hardy-Weinberg parlance, little p and little q are allele frequencies. Little p refers to the frequency of the dominant allele, in this case, big B. So if you take all of those bees from those javelinas, dump them into that Mr. Turtle pool, the proportion of the bees that are big B, that is P. Q is the proportion of bees that are little b. Okay, so those are allele frequencies. So let me say that one more time. If the problem is asking you for the proportion of big B's or little b's, it's asking you for the allele frequencies P and Q respectively. We're only going to be working with two allele genes, so therefore P plus Q has to equal 1 because you can only have big B or little b. So for example, if 60% of the alleles in the javelina gene pool are big B, 0.6, that means 40% or 0.4 must be little b. So that is Q because P plus Q equals 1. All right, so now I want you to think about how we can describe genetic variation. And we're going to look at a population of Gregor Mendel's beloved pea plants here. So we have 9 pea plants with different flower phenotypes. How can we describe the variation that we see in this population? First off, we can think about genotype frequencies. So you have 9 individuals, and if we count 1, 2, 3, 4, 5, 6 out of 9 are homozygous dominant. So the frequency of big W is 0.67. Likewise, 1 out of 9 is heterozygous, or 11%, 0.11, and then 2 out of 9 exhibit the genotype little w, they're homozygous recessive. And if you look, 0.67, 0.11, and 0.22 adds to 1 because there's only 3 genotypes. We can, of course, look at the phenotype frequency. Pretty easy. You've got 9 plants. 7 out of 9, or 78%, show the dominant phenotype. They're purple. And 22%, or 2 out of 9, show the white phenotype. Note 2 different ways to get purple flowers: homozygous dominant as well as the heterozygous genotype. And then finally, we can look at allele frequencies. And this is probably where students get the most confused. But we've got this population of pea plants. There are 9 individuals. Each individual has 2 alleles that comprises their genotype. If we take all of those alleles from these 9 individuals and dump them into a little Mr. Turtle pool, that's our gene pool. And if we count up the dominant alleles, the big purple W's, 13 out of 18, or 72% of the alleles are big W. Again, that is P. P is the frequency of the dominant allele. Likewise, if you count up the little w's, the recessive alleles, 5 out of 18 or 28% are recessive alleles, and that is denoted with Q. Notice P plus Q, 0.72 plus 0.28 equals 1 because it's a two allele system. So, in my experience, the biggest obstacle that students face in doing the Hardy-Weinberg math is that they get confused about what the question is actually asking. So, after you've done the math, take a moment and reread the question. Is it asking you for phenotype frequencies, the proportion of the dominant versus recessive phenotype? Is it asking for genotype frequencies? Or is it asking you for allele frequencies? So you've got to be able to discern the difference. And again, a well-developed cheat sheet will be immensely important on this upcoming assessment.